Hello my dear students, welcome back to Biology at Fujit. Eye development in the Chick embryo is a classic example of embryonic induction, where the optic vesicle, lens and surrounding tissues interact to form a fully functional visual organ. In this video, we will explore the step-by-step formation of the optic cup, lens, retina and associated eye structures in a simple and exam-oriented manner. Introduction. Eye development follows a similar basic pattern in all vertebrates, but the cheek embryo is commonly studied to understand eye morphogenesis. The eye is a highly complex organ formed by the coordinated interaction of different tissues. Eye development is considered an excellent model for studying embryological processes, especially induction and tissue interactions. Major stages of eye development: development of sensory areas, development of lens, development of associated structures, etc. Development of sensory areas, formation of optic placode and optic vesicle. Optic placode: at the anterior end of the neural plate, two oval areas appear on either side of the midline. These areas are covered by ectoderm and underlain by mesoderm. During brain development, the lateral walls of the future diencephalon becomes thickened. These thickened regions are called optic placodes. Optic vesicle: the optic placodes bulge outward laterally to form optic vesicles. Each optic vesicle grows through the surrounding mesenchyme toward the surface ectoderm. The optic vesicle remains connected to the forebrain by a narrow stalk called the optic stalk. Formation of optic cup, lens induction: when the optic vesicle contacts the surface ectoderm, it induces the ectoderm to thicken and form the lens placode. Optic cup formation: as the lens placode invaginates, the optic vesicle also invaginates. This inward folding transforms the optic vesicle into a double-walled optic cup. Choroid fissure: invagination occurs asymmetrically. A groove remains on the ventral side called the choroid fissure. It allows entry of blood vessels and provide passage for optic nerve fibers. Formation of retina and optic nerve. Neural retina: the inner layer of the optic curve differentiates into the light sensitive retina. It develops into 7 layers of nervous tissue. Histogenesis of retina: retinal development occurs in 3 phases. Number 1, cell multiplication; number 2, cell rearrangement and migration; and number 3, final differentiation. Photoreceptors: there are rods and cones. Rods are responsible for dim light vision and cones are responsible for color and bright light vision. Optic nerve formation: fibers from retinal ganglion cells converge toward the optic stalk. These fibers collectively form the optic nerve. Development of lens. Lens placode formation: surface ectoderm in contact with the optic vesicles thickens to form the lens placode. Lens pit and lens vesicle: the lens placode invaginates to form a lens pit. The edges of the pit fuse, forming a closed lens vesicle. Lens fiber formation: cells of the posterior wall elongate greatly. These elongated cells become lens fibers. The cavity inside the lens gradually disappears. Lens epithelium: cells of the anterior wall remain cuboidal. They form the lens epithelium. So important point is, in cheek embryo, lens fibers begin to form around 96 hours of development. Now we'll go for the development of the associated structures. Number 1 is choroid and sclera. Mesenchyme surrounding the optic cup differentiates into sclera and choroid. The inner pigmented and highly vascular layer, that is choroid, it nourishes the retina. Conjunctiva, cornea and aqueous humor. Conjunctiva is formed from the layer between epidermis and sclera. Cornea is formed from outer ectodermal epithelium and inner mesenchymal stroma. Transparency of cornea. Transparency develops due to loss of water or dehydration, suppression of pigmentation and organized arrangement of collagen fibers. Aqueous humor: it's a space develops between lens and cornea called the anterior chamber. It becomes filled with a watery fluid called the aqueous humor. Iris: it is developed from the pigmented retina. It forms a circular curtain in front of the lens. Central opening of the iris is called the pupil. It contains muscles that regulate pupil size. Ciliary body or ciliary muscles, it is formed from pigmented outer layer of optic cup, mesenchymal tissue, and it helps in accommodation of the lens. Vitreous humor, it's the cavity between retina and lens, is called the posterior chamber. It is filled with a gelatinous substance called the vitreous humor. Extraocular muscles, it is developed from condensed head mesenchyme. It is responsible for eye movements. Eyelids, it is derived from the surface ectoderm. The skin folds split into upper and lower eyelids. Its shape varies among the vertebrates. Analysis of eye development, which is the inductive interactions. Eye development depends on a series of inductive events. Number 1, optic vesicle induces lens placode formation. Lens vesicle induces optic cup formation and retinal differentiation. And step 3, optic cup plus lens induce the cornea formation. This mutual interaction is called synergistic induction. Different tissues induce one another sequentially to produce a functional eye. Phases of eye development. Phase of induction. Tissue interactions initiate eye formation. Disturbance prevents eye development. Phase of cell differentiation and axis formation. Cells specialize into retinal cells, lens fibers, etc. Disturbance causes developmental abnormalities. Phase of mechanical tension. It determines final size and shape of the eye. Abnormal tension leads to structural defects. Important experimental finding: optic vesicle expansion caused by pressure of fluid inside the developing brain ventricles. Role of mesenchyme. Surrounding mesenchyme is essential for normal eye development. It helps form supporting structures. Plasticity of optic cup layers. Initially, both layers of the optic cup can change their developmental fate. Lens vesicles induces formation of neural retina. Mesenchyme induces formation of pigmented retina. Role of internal fluid pressure. Amount of pressure influences the proportion of neural and pigmented retina. Corneal formation, it depends on proper tissue arrangement. Mechanical tension, interactions among developing eye tissues. Quick flowchart. Neural plate will form the optic placode, then optic vesicle, then optic cup and optic stalk, then it will form the retina and optic nerve. Surface ectoderm will form the lens placode, then lens pit, lens vesicle, and ultimately lens will form. Mesenchyme will form the sclera and choroid plus corneal stroma and eye muscles. Optic cup will form iris and ciliary body plus retina. Conclusion: the vertebrate eye develops through sequential inductive interactions among the optic vesicle, surface ectoderm and mesenchyme, resulting in the formation of the retina, lens, cornea and associated ocular structures. So my dear students, the eye develops in cheek through a series of coordinated inductive interactions between the optic vesicles, lens and surrounding tissues. These remarkable processes transform simple embryonic structures into a highly specialized visual organ capable of receiving and processing light stimuli. So in conclusion, eye development in cheek is a classic example of embryonic induction where coordinated tissue interactions lead to the formation of complex and functional visual organ. If this class helped you, like the video, share with your friends and subscribe for more biology topics and comment below which chapter you want next. Thank you.